All right, in the previous video, we ended up discussing, or I should say, the last thing we talked about was water movement into and out of living organisms. And the examples we used were simple one-cell animals and one-cell plants. And, technically those are living organisms, but one of the things I did want to discuss a little bit more in depth was kind of some large scale animal osmoregulation. Okay, so this is a new term, osmoregulation, which we'll talk about towards the bottom of this slide. But osmoregulation is actually from chapter 44. This is a chapter we used to cover in high amounts of depth. It is now something that the College Board just kind of wants us to put into another part that actually relates to it. And it turns out that Chapter 7 relates to Chapter 44, or osmoregulation, very, very well. And in fact, I would always cover Chapter 44 directly after Chapter 47. So I've decided to put a few of the tidbits, some of the more important macro-scale things from Chapter 44 into the Chapter 7 lecture here. And the big idea I wanted to center on was this idea of osmoregulation. Okay, so there's going to be terms that I use here that are new and confusing, and you don't necessarily need to worry so much about what they actually are, other than the fact that they kind of relate to stuff that we already know. And so I'm going to talk about what I'm alluding to here as I work my way to the next couple of slides. Okay. So one term I want to discuss right off the bat is called osmolarity. This may be a term that you've discussed in either Honors Chem or AP Chem. I know it's a chemistry term, and what it means is solute concentrations expressed as molarity. Okay, so it's basically what we're getting at here when you hear the term osmolarity is you are thinking of the solute concentration of something. Okay, and we can think of it as all of the solutes kind of in one specific spot. So it could be salt and sugar and potassium. It could be all these different things that are dissolved in the solvent. Okay. Physiological systems of animals operate in a fluid-filled environment. That's one thing we have to know because we take it for granted, but all of the cells of importance to us are actually bathed in what is known as interstitial fluid. Okay. And this is a gross example, but if I were to take a knife, okay, and cut a little patch of skin here, and then kind of fold it back, and if I could do that without damaging any of the blood vessels or capillaries, so that I didn't actually bleed when I did this. In fact, they can actually do this when they do surgery on your hands. So those of you who like to watch surgery videos, you've probably seen interstitial fluid. And they can actually cut off the blood flow up here, and then the digits and things down here don't bleed hardly at all. But anyway, if I were to take that skin and just that layer, pull it back so that you could see all the muscles and things and fat below there, you would see that those structures were bathed in kind of a translucent, watery substance, and that's called interstitial fluid. And in fact, all of the inside of our body is basically covered in this stuff. So this relates to us as well, because our bodies are covered in a liquid environment. The parts that do the functional things for our body are covered in a liquid environment, and many animals and organisms live in water, okay, or have to be around water so that their bodies are constantly wet. So that is why I want to talk about this, because it is relevant. Okay, so relative concentrations of water and solutes within our body have to be maintained within fairly narrow limits. So this is what we know to be called as homeostasis, right? And so everything from our body temperature to our blood sugar levels, even to the concentrations of the solutes in our body, have to be regulated within narrow limits, or else our body's not happy. Okay, and so as human beings, we are considered osmoregulators. Okay, and this means that we regulate solute concentrations. Okay, anytime you hear that term osmo or osmolarity, think solute concentrations. Okay, so osmoregulation allows us to regulate our solute concentrations which then allows us to balance the gain and loss of water between our internal fluids and the external environment, okay? And generally speaking, this is done with the use of some energy in most animals, okay? And we're not going to get into the specifics of energy use and not in these next couple of slides, but I just want to outline a couple of examples. And we're going to jump right to an example here, a couple of examples with two different types of fish, okay? We've got a fish here that is a saltwater fish, and we've got a fish here that is a freshwater fish, all right? And we've got to take into account that these fish are surrounded by a liquidy medium, right? That they live in water of some variety, and that water, depending on where they live, in this case the saltwater guy, is going to have a certain amount of solute in it compared to this animal's body. Okay, now this is always a fun part for me in classes, just to pick your brain about what you see and what this diagram is telling you based on what we already know, what we've covered in the previous videos. Okay, so let me just take a snapshot of this because I'm going to draw on it. Yeah, that's stupid face right there. Okay. Anyway, is that what they call that? A derp face? A derpy face? I don't know. That's what it looks like to me anyway. Anyhow, let's kind of break down this picture. All right, so we are looking at a saltwater fish, and we're going to talk about how this thing osmoregulates when it lives in salt water, right? Because we know that some animals like salt water and some don't, right? So let's see how this guy deals with salt water, okay? Now, the statement up at the top of green says hypoosmotic to seawater. So that tells us some things about him and the seawater, okay? So what does it tell us? What does it tell us about the fish's internal solute concentration as compared to the saltwater, the surrounding seawater? So pause it and answer that question. What does this term up here, or this statement about the fact that that fish is hypoosmotic to the seawater, what does that tell us about his internal solute concentration? Okay, if you said that it is lower amounts of solute than the surrounding environment, you would be right, okay? So this guy actually has less solute in his body as compared to the seawater. And that makes sense, right? The seawater has a ton of salt in it, right? And so the solute concentration of the saltwater should be relatively high because there's a lot of solute dissolved in it, being salt. And it's going to be more, okay, than his body has. Okay, so let's think about what this does. Now, his body is able to regulate levels of not only solute but water. Okay, so let's look at these arrows and decide what they're trying to tell us. These blue arrows. I will ask you a question, and you pause it and see if you can get it right. Would he gain or lose water living in the situation that he does? Would he gain or lose water living in the situation he does? Okay, theoretically, if all things were equal, he would lose water because we know how water moves, right? Water moves from areas that are hypotonic, okay, which is him, to areas that are hypertonic, which is the seawater. So water would evacuate from his body, which is what we see happening right here. Okay? Now, or we could also say it as water likes to dilute the solute. Okay, so there's more solute out here, okay, with him than there is inside of him, so water will leave him to dilute the surrounding environment. Now, we can ask ourselves this next question. Do we see massive amounts of shriveled up fish on the beach, all dead? No, we don't. So obviously this guy is doing something that allows him to continue his normal life, right? And so what he's got to do is he's got to balance this water loss. So how does he do it? Well, like anybody else who has a lot of water loss. If you go work out for 2.5 hours very strenuously, you'll be at a loss for water. What do? You drink water, right? Okay, so that makes sense, right? So that's why we've got this arrow here, because he drinks a lot of water to replace the water that is lost here. He is a fish, as we know, right? And as a fish, he has got plenty of access to water. So he's just going to drink water. And it'll replace, even though he's drinking salt and stuff like that, it's going to replace the amount of water that he's lost just by osmosis, because naturally his body is going to want to lose that water. Okay? Now, something else that we do as humans, if we have too much water, what do? We excrete it, right? We go to the bathroom and the toilet, we get rid of excess water. This guy doesn't really have that issue, and as a result, he creates what is called scanty urine. Okay? This is another adaptation that allows him to combat this water loss. Okay? He produces scanty urine. And scanty urine it's gross. It's basically like a paste. You know, I tried at one point to find, because I think I watched some nature documentary of a shark peeing in the water or something like that. And I tried to search for a video of a shark peeing in water, and apparently YouTube doesn't have a whole lot of people who want to videotape sharks peeing in the water. But I seem to remember this, and it came out, and it turned the water like, it was like cloudy, right? It was almost like a paste, and when it hit the water, it was like cloudy. And so that's what this guy would do, living in the salt water. He wants to conserve as much water as he can in his body, so he's going to put very little water in his urine. Okay, and small amounts, very, very small amounts of water. So this scanty urine is almost like a paste. Can you imagine peeing paste? That's gross, right? This guy kind of does that. That's one of his adaptations, and that allows him to keep this as much water in his body as he can. Okay, so he's got a couple of adaptations that allow him to hang on to his water as best he can, or replace it as best he can. Now let me replace what I had just. He is low in solute, and this is high in solute. Okay. The other thing that we have to remember here is that this is not an ideal lab situation. This is real life. This is a fish that lives in the environment, and water is not going to be the only thing that moves. Okay, so we talked about the osmotic loss of water, right? What else is going to move across his cell membranes? Okay, this is a question I want you to pause and try to answer. What else is going to move across his cell membranes? If you answered salt, you were correct. Okay? We have two concentration gradients here going on. We have the concentration gradient of the water. Okay, so it's a lot more water in here than there is out here, so water is going to leave. But we also have the concentration of solute. Remember that there is solute here, okay, stuff dissolved in the water, and we have a certain amount of solute here. Okay? Now remember how things move down their concentration gradients. They go from areas that are highly concentrated, right here, to areas that are low concentrated, which is inside the fish. So we actually have another diffusion occurring here. We have another concentration gradient where salt is actually going to come into this guy's body. Okay? So he's going to get extra salt in his body, which he doesn't want there. That's not within his set limits, within his normal limits. And we want to make sure that, or he wants to make sure that he can get some of that extra salt out there, out of his body. And so what does he do? What are his adaptations for that? Again, we've got this urine, okay? So not only does it double as a way, not only does his urine double as a way for him to maintain a certain amount of water or keep water in his body, but allows him to excrete extra salt ions that he takes in just passively, naturally, okay? In addition, his gills are very good at excreting salt ions as well. Okay, there's a lot of surface area on those gills, and his gills will just naturally release some of that extra salt that is taken in either through drinking the seawater or through natural diffusion. Okay, so this little guy living in seawater can tell us a lot about the movement of stuff into and out of his body, both water and solute. Okay, let's look at the next guy. The next guy is our freshwater friend, and I'm going to take a snapshot of the freshwater guy here as well. The freshwater guy, and I'll ask you kind of the same questions or similar questions, and you can try to pause the movie and answer. Okay, let's talk about him. He lives in freshwater. Okay, situation number one. and he is hyperosmotic to the fresh water. Okay, so I want you to try to answer which has the higher concentration of solute, the fish's body or the surrounding environment? Okay, if you answered the fish's body, you would be correct, and that means the surrounding environment has less solute. Think about it. Fresh water, it probably does have some solutes in there, okay, some stuff dissolved in there, but probably not nearly as much as the cells in his body. Okay, so what does that do for him? What is he going to experience in his everyday life? Okay, based on just natural passive events, okay, and the movement of stuff along concentration gradients, he will actually gain water. Water will go into his body at a fairly good clip. Now we've got to ask ourselves, do we go to the river or the lake or the pond and see 1,000 bloated fish on the side all dead because they've puffed up with water and died? No, we don't see that, right? So obviously they've got some adaptations that help them to deal with this situation. That allows them to osmoregulate in a certain way, regulate the amount of water and solute in their bodies, right? Okay, so let's talk about his adaptations, all right? Number one, he excretes large amounts of dilute urine. Okay, this is the same thing as you go to the gym and the big meathead guy has 4 gallons of water that he drinks the entire thing during his workout, right? Or the people walking down the hallway when we used to go to school. You remember that? That was like 15 years ago, right? They drink water all day and then they go to the bathroom every 10 minutes. Hey, Mr. Sonic, I'll go to the bathroom. No, you can't. You pee your pants. So that's the same situation as this guy. He's taking in water just hand over fist and just can't get enough water passively. That's just what his body's going to do. It's going to suck in water because water moves from areas of hypotonicity, okay, which is this area here. The environment is hypotonic, and his body is hypertonic. So water moves from areas that are hypotonic to hypertonic, or water will dilute the solute, right? And so his body is just naturally going to want to take water in, so he just pees it out. Okay, it's a really, really easy adaptation for him to have. Okay, in addition to that, he can kind of change the internal concentration by taking in some more salt ions through his food. He can also take on some more salt ions through his gills. Okay, so that's the water situation for the freshwater fish. Let's talk about the solute situation, because remember we established in the last slide that there is actually 2 concentration gradients, or there are 2 concentration gradients in play here. The one for water and then the one for the solutes. Okay, so what's going to happen with the solute one? Remember that solutes will move from areas that are more highly concentrated to areas that are less concentrated, okay, in an attempt to reach equilibrium. Okay, so this guy is naturally also going to just lose salt or solutes into his environment, so he's got to figure out a way to replace those. Okay, so just by natural eating, okay, eating food, he's going to take in some of those salts, and he can naturally passively take in salt ions through his gills just by keeping his gills open to the water. Okay, so he can actually replace these solutes that are lost to the environment in fairly passive ways. Okay, so I hope you did learn a little bit here from our fishy friends. It's kind of a fun Q&A, question and answer type of thing that gets you actively thinking about which way solutes and water will move under given situations in living things. Okay, we have similar adaptations, though far more advanced. Our kidney is the organ in our body that helps us undergo this osmoregulation. Okay, this is a diagram of a part of a kidney called the nephron, and the nephron is responsible for actively filtering our blood and also helping us to maintain water or keep water in our bodies, but also keep valuable solutes that we think we need. Okay, and so this is a diagram of the nephron. See. Pardon me, this is a diagram of the nephron, and this is the tubule system in our nephron that allows us to be able to keep as much water as we can. Now, this nephron works by creating concentration gradients. Now, in previous classes, I would have spent a day going through these couple diagrams that you've seen here. But basically what you're seeing here, and if you want to kind of take a look at the numbers on the side, the numbers indicate the concentration of like solute that is in the interstitial fluid that bathes this entire part of your body, okay? And so it's more highly concentrated down here than it is at the top. And you'll notice that there are some subtle differences between the numbers inside the tubules as compared to the numbers outside sometimes, okay? And so what this actually does is it allows for your body to just passively draw water out of the urine, okay? So this stuff in blue is ultimately going to become your urine. And your kidneys allow you to draw water back into your body to save if you need it, okay? It also allows you to save certain things like salts, maybe some sugars that got filtered that shouldn't have. So our body has this really cool adaptation called the kidney, and at the smaller level, the nephron, that allows us to do essentially the same thing as the fish. It allows us to keep our bodies within certain set limits of solute and also water, okay? So if you do want to know more about this, we'd have to set aside a 30-minute talk, and we don't need to, okay? The College Board has kind of taken this out of our hands. We don't really need to cover it anymore, okay? So I wanted to give you an overview of how different organisms utilize these processes, diffusion and osmosis, to maintain their osmoregulatory functions, to maintain these certain set limits of solute and water in our bodies, okay? The last little bit of information here I want to talk about is going to deal with different membrane transports and transport proteins and different ways of transport across the membrane. I know we've talked already in a couple of the videos about the movement of stuff across membranes, but I want to make sure that you understand that we can discuss this topic in more broad terms as well, okay? And then we can add more to the picture, because there's always more to the picture, and that's what we're going to do here in the last few slides. Now, this video right here, I think it works if you guys are able to access it, and I will provide the link. And it describes all the different ways of transport and different types of transport proteins that our body has. And I think it's a good transition between what we've already covered and what I'm going to cover in these last few slides. So before, maybe if you want to pause the video here, and then you can access this video, watch it, and then we can you can watch the second part of this video here, okay? So the first thing I want to talk about is passive transport. And we've already kind of discussed passive transport without really knowing it. And maybe it's something that I should have introduced first instead of last. It is what it is. You get the information regardless, okay? Passive processes happen when no energy is used in the process, okay? So when we think about things that happen in the cell, think about things that are passive. They don't require energy to happen, okay? Some examples of processes that we've already talked about that are passive are simple diffusion and osmosis. Those things happen because there is potential energy that's been built up on one side of a membrane or something of that nature, and that potential energy is released when there is a movement. And when that potential energy is released, it is generally considered a spontaneous process, and spontaneous processes don't require energy, okay? So simple diffusion, the movement of something just directly across a membrane, okay, is an example of this. Osmosis is an example of this, where water goes from a place where it's highly concentrated to an area where it's less concentrated. Or when food dye goes from an area that's more highly concentrated to an area that's less highly concentrated. Or when a student crop dusts my desk when they walk by, you will all eventually smell that. Maybe not with masks on and everything, but it's more highly concentrated at my desk, and then it eventually goes throughout the rest of the room, okay? Now, there is also a process known as facilitated diffusion, okay, which is distinctly different from simple diffusion, okay? Facilitated diffusion is the diffusion of solutes with the help of transport proteins. And if you remember what transport proteins are, they are little proteins that are embedded right into the cell membrane, and they allow hydrophilic channels for molecules to pass through. Okay, so the term to facilitate means to help. Okay, so I am facilitating your learning right now. I'm helping your learning, hopefully. And some molecules require the help of proteins to pass through the lipid bilayer. And we've touched on this tangentially already a little bit. Because these molecules are either two polar or ionic, they can't pass through the highly nonpolar cell membrane. Okay? So they need help. They need basically a pathway. They need a tunnel. And that's what these transport proteins do. And we call this facilitated diffusion. There are a few things that can transport over the cell membrane by way of simple diffusion. Things like carbon dioxide, things like just molecular oxygen. Those are nonpolar molecules, and as a result, they can transfer simply just through the cell membrane without the use of a protein, without the use of a channel. Okay? Active transport is the opposite of passive transport. Active transport is the use of energy to get one thing from one place to another. Okay? And there is a reason that you have to use energy to get one thing from one place to another. You have to work against the concentration gradient. And I will show you a picture that I hope will help you understand this, okay? So in active transport, you generally have things called pumps that will actually take molecules and put them where they shouldn't be naturally, okay, or passively, right? And this requires energy to do that. And this is used to maintain something known as the ionic gradient. And these ionic gradients represent potential energy. Okay, so there are instances in our lives and in our bodies that our cells will want to spend energy just to pump substances to a place where they shouldn't be. Okay, and so when we are, go up here, take a picture of this and then I'll write on it. All right, so if I have a cell membrane here, what active transport will do, this is a substance on this side, and this is the same substance on the other side. What active transport will do is spend energy. I'll use this guy. That looks like urine. Okay, we'll use this. We'll spend energy to move this particle to the other side. That is against what it would naturally want to do. Naturally, okay, passively, these molecules are going to want to do this. They're going to want to go down their concentration gradient, okay? That's what they naturally should do. That's their natural state of things. What the cell sometimes will do, will spend energy to send them against what they would normally want to do. Okay, so it will spend this ATP. Every time it'll do that, it'll spend an ATP and put extra stuff out here. So we're increasing the concentration. There's now going to be lots of this stuff out here and very little of this stuff in here. Okay? So we'll talk briefly about why the cell would actually want to do that, but I wanted you to see that before we go on to the next thing. And so when you've got this big stockpile of stuff on one side of the membrane, what that represents is potential energy. Okay, and I've discussed and talked about the Hoover Dam before because I think the Hoover Dam is a really, really good analogy for all of this stuff, right? So the Hoover Dam itself, let me take another picture here. The Hoover Dam itself would be like the cell membrane. Okay. The cell membrane, in some instances, has got some kind of separation. And up here we see we've got a large amount of, we'll say, solute, and there's a less amount of solute down here. So naturally, the water, in this case, is going to want to flow through the Hoover Dam, does it not? And it will. And as a result, when it does that, this up here has potential energy, and when it flows through the dam, that potential energy is harnessed, and we get electricity from the dam. Okay? All right. Now, there are some instances where our cells are going to go against what would naturally happen, okay? And so active transport it's as if you had like a little pump, like a little water pump down here, and it runs off gasoline, okay? And we run like a hose back up here to the reservoir, okay? That's a little hose up to the reservoir. It would take a lot of energy. You'd have to fill this little gasoline water pump with gasoline a bunch of times, turn it on, and it would pump water up the Hoover Dam and put it back here. And so that's what our cell does from time to time. It will actually take stuff from here where there's less of it, put it back up here where there's lots of it, and as a result, you've got to spend some energy to do that, right? Or think about it this way. If you wanted to do this by hand, you go down here, get a bucket, okay? You walk up the hill, you dump it back in the reservoir, right? That's a lot of energy you just wasted to take one bucket of water all the way up here and put it up here where there's a ton of water already, right? So this is active transport. That is active transport, and our cell will do it. Okay, and it was one thing that you can kind of rest assured that if the cell is going to spend energy on it, it's going to be important either to the cell or the organism as a whole. Okay, now when the active transport occurs, it creates a concentration gradient of an ion generally. Okay, and this is something known as the membrane potential. This causes what is known as the membrane potential, and this is stuff that we'll talk about far into the future, hopefully. And it is called an electrochemical gradient. Okay? Ions have an electric charge. They are also chemicals, which is where this name comes from, the electrochemical gradient. Now, one thing that ions also do, they move down their electrochemical gradients as well. So it seems counterintuitive. All right, Sutherland, you just told me that our cells spend a lot of energy to move ions from places where there's not very many of them to a place where there's a ton of them already. And then you're telling me on the slide right after that those ions just come right back down. That's exactly what I'm telling you, okay? Because this movement of ions back against the concentration gradient isn't going to happen naturally. The cell does have to put some energy into it, which means that these processes are actually vitally important to the living of the cell or the living of the organism. Because when ions move down their concentration gradient or their electrochemical gradient, what we actually get is an electrical voltage. We actually get nerve impulses. Okay, so the fact that we've got this process here, which drives our nerve impulses, I bet you're glad that your cells waste some energy to do that, right? Because otherwise you don't interact with the environment, you don't interpret what's going on around you, and it'd be a really bad existence, right? And so this maintaining of the membrane potential or the electrochemical gradient, which the cell spends energy on, is vitally important to living organisms for the reasons I've talked about there. Okay, so there are certain types of protein transports or transport proteins that will help to move these ions from down below the Hoover Dam to up on top of the Hoover Dam. Okay, and they are called sodium-potassium pumps. This is a main example of one, and I think this works. I tried it out. I'm going to try to see if it'll work this time. It's a flash-based thing. We're going to allow it, and I'm going to let you listen to what she has to say.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here is how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, providing the energy that causes the protein to change shape and push the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
So what we see in this picture right here is the establishment of two different electrochemical gradients. And eventually these things, these K pluses and these Na pluses will move back to the other side. They will diffuse back to the other side. And we're not going to get into why that is, but it's part of the nerve impulse. Okay, so the K plus will go to this side, and then the Na plus will diffuse back through this side. And as a result, we're able to properly conduct nerve impulses. Okay, so you see this little transport protein was responsible for reestablishing this ionic gradient that didn't naturally exist before. Okay, had to spend some energy to do so. That's why it is so important. Okay, so this diagram is kind of a walkthrough, gives you step-by-step exactly how that works. All right, so this creates what is known as the membrane potential, which is a voltage across the membrane. Okay, when we separate charge, I don't know a whole lot about electricity, but I know that when you have a separation of charge, you get a voltage. So there's actually a small voltage. If you were to put a voltmeter on one side of the cell and then on the environment part of the cell, so you got the internal part of the cell and then the environment, the voltmeter would actually read a small voltage at around negative 70 millivolts. And the reason for that is because your sodium-potassium membrane pumps have established some separation of charge. Okay, so that's part of it. There's an unequal distribution of charge. There's also an unequal distribution of anions and big proteins that are negatively charged inside the cell versus cations outside of the cell. Okay, so these electrogenic pumps, the sodium-potassium pumps, help to generate that voltage across the cell membrane. Okay. Okay. We've already been introduced to a proton pump. Okay, this is another type of protein that will help us to move things across the membrane. We talked about how the lysosome needs an internal pH to be acidic, and in some instances we'd have a proton pump that would actually pump protons. And protons are nothing more than H plus molecules, because that's all it is just a proton. It's just one proton in the nucleus. There's no electrons left over. But generally speaking, if the cell wants to create an acidic environment somewhere, let's say for hydrolytic enzymes to work properly, then it would use a proton pump, and it would pay a little bit of energy to do so. Okay, so that's one option for transport across the membrane as well. Co-transport is another option of transport across the membrane. So what you see in this last section that we've been discussing is we're kind of branching off from simple diffusion, which we talked about earlier. Okay, so co-transport is something that's a nice option to have for cells to get big molecules into their bodies or into their cell bodies for relatively cheap. And oftentimes it is coupled with something else, like in this case a proton pump is going to spend some energy to get a proton out, and maybe the sole reason it wants the proton out is because that little proton can actually go back through this little protein here called the sucrose H plus co-transporter. Cool, right? It can go back through and interact with this protein, and the interaction of this proton with this protein will allow sucrose to hitch a ride. Okay? And so that sucrose molecule will come through along with the H plus molecule, and basically what it's doing is it's harnessing this energy that was used before. Okay, so we invested in energy here to get an H plus out. That H plus's interaction with this protein right here is going to allow sucrose to come on through. Okay, so it's a kind of a roundabout way to get sometimes big molecules into the actual cell. Okay. The cell can also transport very large molecules through two or three different types of processes. Okay, one, the general term for importing things from outside the cell is called endocytosis. Okay, so if a cell wants to take a big thing that's outside of the cell and bring it inside, it's going to undergo endocytosis. Okay, endo sounds like into, endo, into, endo, into. Endocytosis brings things into the cell. Okay, generally big things. Phagocytosis is the specific term that describes the cell wanting to bring large particulate substances into its body. In other words, phagocytosis is the cell's ability to eat. Okay, it's eating food. Eating food. So phagocytosis is eating food. All right. Penocytosis is a word that describes endocytosis of fluid and things dissolved in fluid. Okay, so you can think of this as the cell drinking. So penocytosis is the cell drinking. Phagocytosis is the cell eating food. Okay, there's a third type of endocytosis called receptor-mediated endocytosis, and this is basically just like a way for the cell to bring something into its body and making sure that it is safe. Okay. And this is mediated by receptors on the surface of the cell that can identify things as either harmful or not harmful. And if it's not harmful, it will take it in. So these little receptors identify things that are good for the cell. Okay. The other way of transport of large molecules is by exocytosis. Exocytosis is the dumping of vesicles given off by the Golgi body. An example of this would be insulin from pancreas. Another example would be waste. Okay, if the cell has waste that it wants to get rid of, it will do so by endocytosis. And so if you think of the exocytosis. If you think of endocytosis as the cell eating, you could think of exocytosis as the cell taking a dump. Okay. Exo sounds like exit, exo, exit, exo, exit. Exocytosis is things exiting the cell. Okay, oftentimes it can be waste, but more often than not, it is some kind of product that the cell made, like the example given here is insulin. Okay, so some vesicle will come. So if this is the cell membrane, some vesicle will come from inside of the cell. That's over here. It'll fuse with the cell membrane and it will dump that stuff out into the environment, which is out here. That's kind of handy. Okay. So I think that finally does it for all of the official material that we were going to cover in Chapter 7. This is a lot. It's 4 videos. Each one's at least 30 minutes long. Spend time going back and reviewing, rewatching the things that you didn't understand. Jot down notes. Make sure that you are taking notes along with this, because I say a lot of things that aren't necessarily in the lecture itself. So be an active learner, be an active listener, and go back and review things if needed. Okay. So enjoy, and I'll talk to you soon. Have a good one, guys.